Thank you very much for the invitation to present at the Cell Biology 2021 event. Today I will be taking you through the path from basic research in cell biology, where we actually measure and study specific mechanisms of disease in oncology, and moving towards actually detection of these characteristics in blood using blood-derived exosomes, extracellular vesicles that are being shed by cells. So this is the path that we actually take. Of course, we start off with basic research where we study the cellular biology and the mechanism of disease. And once we have identified specific biomarkers that will give us information that can predict disease or on therapy outcome, or actually biomarkers that can be used to monitor patients, then we trigger immediately the path towards validating these biomarkers using patient material, patient data, and with all this information we will translate into a pipeline, a workflow, in which we can actually then measure these biomarkers depending on the specific question. So ultimately we will validate these diagnostic or theranostic biomarkers towards eventual commercialization. Now, our group have been studying the particular phosphatase, the phosphatase protein 2A, PP2A. PP2A is a phosphatase complex made up of three subunits. You have the A structural subunit, then you have the B, which is a regulatory subunit, and the C subunit, which is the catalytic subunit. And this complex is directed towards specific targets. In this case, I'm showing that the targets are actually the PI3K ACT mTOR pathway, and they dephosphorylate these targets. Now, when the complex is inhibited, this particular mechanism, this particular signal transduction is sustained and promotes survival, proliferation, and also differentiation block. Now, previous studies we showed that specific proteins, these are actually regulators, so it's one of these B subunits, but in this case these are inhibitory regulators of this particular complex. And our journey started with actually identifying the role of alpha-4, or also known as IgBP1, in shifting the phosphatase activity towards different targets, but away from the mTOR pathway, resulting in a differentiation block that was shown on an erythroid progenitor model. Today we know a lot about these inhibitory subunits. Alpha-4 is one of them. SET has been shown to be extremely important in blast crisis in chronic myeloid leukemia. And CYP2A has gained a lot of interest in solid tumors. And this is where we are currently working with CYP2A by overexpressing it in various cell lines and studying the feedback mechanisms in various cellular models. In fact, we have now at hand cellular models which are breast cancer cell lines in which we constitutively expressed CYP2A, and we looked into the phosphoprofiling due to the effect of this overexpression. So the importance of PP2A in breast cancer led us to looking at the sensitivity of cell lines. In this case, I'm showing the triple negative breast cancer cell lines and their sensitivity to PP2A activator FTY720. Now, as you can see in these graphs, so the graph A shows TMBC cell lines that are sensitive to FTY720. There is the percent cell viability on the y-axis, and on the x-axis we find the concentration of FTY720. And you can actually appreciate that these three cell lines in figure A show that you can actually reach the IC50 at very low dosages of FTY720, two of which even less than 1 micromolar, whilst the TMBC cell lines in figure B show that they are not sensitive to FTY720, with one of the cell lines showing that there is a change, or there is an IC50 reached at 10 micromolar FTY720. So basically here we can see that there are TMBC cell lines that are sensitive and those that are non-sensitive, and actually we studied further these 6 cell lines as 2 groups. So we can see that we have the NDAMB231 reaches an IC50 at 1 micromolar, BT20 at very low dosages of 0.1 micromolar, and HS570 at 5 micromolar. And we compared these 2 cell lines using transcriptomic datasets. So we did RNA sequencing experiments to look at differential gene expressions between these 2 groups. And from these transcriptomic analysis, we identified a list of potential markers that are associated with sensitive cell lines to FTY720. Our understanding was, or our hypothesis was, that these markers are actually markers of low PP2A activity, and hence we continued with the experiments showing, and we actually showed that these sensitive cell lines have very low PP2A activity, which I will show in a minute, as compared to the non-sensitive. And then we utilize these differentially expressed genes into multiplex panels to compare these cell lines and even move on to patient material. So as part of the multiplex panel that we actually selected, we added the subunits of PP2A, and here we can see that we added the PP2CA, which is the catalytic subunit, and the blue boxes. Then we have also regulatory subunits, the R2A, which is the light blue, and also the inhibitors SET binding protein 1, CYP2A, and AMP32A. And when we look at this data set, in which we are here, the figure shows normalized expression to housekeeping genes, and compare as a group of cell lines the non-sensitive to sensitive, the distribution of the PP2A complex and its regulators, we have a slight significance of CYP2A high in the sensitive group. But this data shows us that it is extremely difficult to compare the markers at group level, so the sensitive as a group and the non-sensitive as a group. I'm saying this because if you then look at specific cell lines, we can see that the 231 breast cancer cell line actually has a very high expression of CYP2A, while in the case of BT20 we see a very low level of the R2A subunit, meaning that different cell lines would have different deregulation parameters for the PP2A complex. Hence, this shows us that it's extremely important to look at the different cell lines, and also when we look at a group of cell lines and eventually patient material, we definitely would need to have a multiplex panel in which we have all the subunits that show the complex, but also addition of a panel of markers that show low PP2A activity, and in fact that is what we were after. And therefore we moved on to multiplexing, and we used this Quantigene plex assay from Thermo, which works on the xMAP technology using the Luminex platforms. So the Luminex xMAP bead-based multiplexing can go up to 80 genes. In this particular experiment we started with 40 genes. It is a nucleic acid sandwich hybridization method in which you have your target that you want to capture. Then you would have different capture beads, specific probes and capture extenders, which would specifically bind to one of the targets of interest. Now, once it is bound to this bead, then through label extenders, a DNA scaffold is used, in which, using a preamplifier and amplifiers, you will have this whole scaffold of label probes, which would eventually, through streptavidin PE, would eventually give us a signal. So basically here what we're doing is we are not amplifying the target as we do with PCR. Here we actually amplify the signal. And then, using the Luminex platforms, we measure the fluorescence. Now, why we wanted to go for multiplexing? It's basically because cell biology is dynamic, cell biology is complex. And when you look at some of the cell lines that we used, and here we divide them as primary cell lines and metastatic cell lines, those that are sensitive to FTY720 are marked with an asterisk here. Okay. And you can already see that this sensitivity is not based on the expression of epithelial markers, because here you see ecadherin and EpCAM. This is the median fluorescent intensity that we get from the Luminex, so this is normalized to housekeeping genes. And here you can see that these are epithelial because you have expression of ecadherin and EpCAM, whilst the others are not. So this is only showing 2 of the plex. Then we looked at, this is the same graph, but now I'm extending the scale here. And here you see, like, if we look at specific mesenchymal markers like encadherin, SNI1, SNI2, FN1, and twist, you can see that now this shows up as a mesenchymal marker cell line, and that is sensitive to FTY720. When we look at vimentin, which is another mesenchymal marker, you see that this is positive for vimentin. So this is always the same, but we are scaling it up to see the markers. So this shows the importance of actually having multiplexing, so that you actually have a good classification. So these are called classifiers of the different cell line models that we use, using only one marker or 2 that would not provide us with the full information. So coming back to our interest to show the low PP2A activity, what we did is we compared these groups at a transcriptomic level, and we identified differentially expressed genes between sensitive and non-sensitive. Here on the left you see a graph in which we actually pulled down the complex and measured the activity, the phosphatase activity, against a substrate in vitro. And as you can see, the sensitive have low PP2A activity and the non-sensitive have a high, but this is something we cannot do on patient material. Of course, we can do it on cell lines, but not really on patient material, and therefore we wanted to find this marker that discriminates between these activities. Here we just add this as a positive control because we know that K562 have actually low phosphatase activity. So when we did all this transcriptomic and checked the networks and gene networks, we encountered this particular panel, and we put our interest in AURKA kinase A and KIF2C as the markers. When they are highly expressed, then we always have a low PP2A activities in our cell lines. And of course we extended studies with inhibitors and activators, and we looked at all the possible designs to show that AURKA and KIF2C are really excellent markers for low PP2A activity. And you can see it here. Okay, so AURKA, these are non-sensitive and sensitive, and this is the activity, corresponding activity. So then once we actually found this marker, we said, okay, how important this marker is in the context of breast cancer patient tissue. And here where we started using the publicly available data sets, in this case the TCGA, and here we're looking at RNA sequencing normalized expression values, looking at matched normal to tumor. So we only looked at those samples in which they have matched normal and tumor, and we compared the expression of AURKA and KIF2C, AURKA in yellow and KIF2C in red. They seem to move always together, but the most important is that there is an increase in these markers when comparing normal to tumors. So that was already very interesting to see. More interesting is that you have a higher magnitude in luminal B, which is known to be the most aggressive, and high in Ki-67 proliferation marker, and also in basal. So this is very interesting to see that we actually have markers that shows us a difference between tumor and matched normal in the different subtypes. Of course, we also did a lot of work on our TNBC patient material, and to our surprise, AURKA is expressed at a high level in more than 72% of TNBC patients. Here we used immunohistochemistry on FFPE whole sections. We did the H scoring, and we used 160 cases for this particular experiment. And here you can see the increase in the H scores of AURKA kinase A, or AURKA, in the TNBC patients. Now, we kept on looking at some important data sets as well, and here again we move on to the TCGA dataset. This is something that we've seen from our FFPE material, where we saw a correlation between the expression of AURKA and one of the inhibitors of PP2A in breast cancer patient, and this was a correlation between AURKA and CYP2A. And here we extended this to also TCGA datasets, where we show here in the left that AURKA messenger RNA expression correlates with the expression of CYP2A, which is quite significant. Now, it's a well-known fact as well that AURKA is amplified in various cancers. And in fact, the AURKA copy number variation correlates excellently with the TPX2 gene. That's because they are on the same chromosomal region and they are in generally amplified together. I'm saying this because we did a lot of work on gene amplifications in various cancers. There's a high frequency of these amplifications, and today we know a lot about gene amplifications. So we started this project. What we did is Again, we wanted to go for multiplexing, so not really studying one, so we do a simultaneous analysis of various gene signatures, and during the experiment we used even 40 plexes. We looked at these common amplified genes in CRC within an 18 multiplex assay. We looked at those that are involved in metastasis and invasiveness, and also therapeutic resistance. This is a specific cohort of CRCs, and this is patient cohort of CRCs, both primary tumors and matched biopsies from metastatic lesions. So it's really a very specific cohort, meaning that these have already had a metastasis, but we went back to the primary tumor, and also we actually had the metastatic lesion, both in FFPE, so formalin fixed, paraffin embedded. It's good to say that with the Quantigene assay we can actually run FFPEs, because it's not you can actually read these gene amplifications and gene expressions in this case using even degraded material. So here we show that there's a huge amount of TPX2 represented in this particular group of patients. So TPX2 was overexpressed at an mRNA level, okay, which is for us reflects that there is a gene amplification. So overexpressed compared to normal colon. Then if you take TPX2, FZD1 and IGF2, we have all the cohort together. So that's a nice panel for these specific types of tumors. Then we looked at the metastatic lesions, where we find 50% of CRCs moving to the lung, showed a very high expression of TOP2A. CCND3 cyclin D3 was overexpressed in lung metastasis. Those that are exclusively found in liver metastasis, CDX2 and KIT in this case, and MYC was found consistently gained in metastasis over the primaries in all the types. And then, okay, so we had sort of an idea of the gene amplifications at primary and metastatic, and then I wanted to move on with other projects to really look at innovative molecular profiling assays, to look at distinct circulating tumor cells and also exosomes. We wanted to develop an actionable panel, multiplex panel, for these circulating cells or vesicles. Okay, so I'm not going to take a lot of time here. So liquid biopsy is you have a solid tumor and these will shed exosomes. You have to have, there is also apoptotic bodies, DNA, RNA, okay, cell-free RNA, and CTCs. So these are all the different ways how we can measure it. We focused on CTCs, but in this particular presentation I will focus on exosomes. Now, in general, exosomes are used to read microRNAs. In our case, we used it to read messenger RNA expression to reflect gene amplifications which originate in the primary tumors. CTCs, as you know, can be pulled down with EpCAM antibodies, although in our research we also showed that some of them are EpCAM negative. So here again, putting multiple antibodies would be necessary depending on the patient. And first we wanted to look again to the amplifications in CRCs. So we have cell lines, we have blocks, so we have a whole workflow in which we can actually do IHCs, look at markers, at histochemistry. For instance, this is an SKBR, which is HER2-positive cytokeratin 19. And this we wanted to really look how sensitive our technology is, so that we move on to exosome research. For instance, if we use EpCAM, we will never capture such cells like the HS5780 because it is EpCAM negative. So we managed to detect amplifications as expected in cell lines. For instance, if we look at SW48, we know that there are the MET and the GFR amplifications. We see them at a cellular level in the cell line itself. That's normal. Okay, but then our interest was to actually see these reflected in the exosomes from which that are generated from these cells. So we started off with another plex. Here we have a 6-plex looking at CTCs. We have EpCAM cytokeratin 19. We have FN1. This is a marker that we really work a lot on, and we've shown that this is involved in early invasiveness. So then we had HER2 there, ER, because we had breast cancer cell lines for this particular study, and MUC1, and we use Luminex technology to read these. Now, what's interesting here, we did a titration experiment with different cells. So here you see the MFI, mean fluorescence intensity against cell number. Okay, and here we have blue is EpCAM, orange is cytokeratin 19, yellow is FN1, and green is HER2. Now, what we can see here, if we look at SKBR, which is HER2 positive, we can see that the green one, HER2, okay, is very high, cytokeratin is very high, EpCAM is high, and it is negative for FN1. So if we have FN1, we don't find these. If we use only EpCAM, you can see that the magnitude is not as high as using HER2 and cytokeratin 19. So in this case, we of course promote the use of cytokeratin 19 and HER2 for this particular cell line, because we can actually detect down to 5 cells here with this Quantigene assay. Whilst using this cell line, we can see that again EpCAM is high, but you don't detect lower than 40 cells. Was with HER2 and cytokeratin 19, we can go down to 15 cells. That all depends on the expression level of these markers. Now look at this. This is totally different. HS5780 is negative for everything except FN1. So and with FN1 we can even read 5 cells. So the importance of multiplexing in actually detecting these CTCs. And then, okay, we moved on to exosomes, and here is where one of our industrial partners, Quantigene, is working and helping us with, is to put together a workflow, automated workflow with microfluidic miniaturizations to increase the sensitivity, so that we actually read the exosomes and plasma by immunoprecipitating them with specific beads. So the exosomes are these membrane-bound extracellular vesicles from endosomal origin. Here you can see electron microscopy of these exosomes. Keep in mind that exosomes are extremely small, so 30 to 120 nanometers. If you have something which is in the sort of 2 microns, those are apoptotic bodies. And in general, microvesicles are sort of indicating all these types, but we are working with exosomes. Now, exosomes are from endosomes, so they have specific markers. So mainly we worked with CD63 and CD9. Yeah, we have streptavidin donor beads. We biotinylate antibody which would bind to exosomes. In this case, it's CD9. Then we have, of course, the exosome capture complex. And from this exosome capture process, we are enriching the exosomes from plasma. Okay, and once they are captured, then we can also see these captured exosomes in flow cytometry. Okay, using, for instance, an CD63, which is conjugated with FITC. And this is the exosome detection complex that we see on flow cytometry. And it's easy to put together. Okay, you can look at CD63, FITC. You can also use other markers like HER2 if it is a HER2 positive cell. And this is what you see on a flow cytometry. You would have these single beads which are loaded with exosomes. You will see the double beads and the triple beads, but even less and less every time. So we can gate on these single beads, and we can see actually the positivity of these exosomes to CD63 when we spike these exosomes in blood. Okay, so here we can see a shift. This is the no exosome control against the exosome sample. So this is HS5780 produces exosomes. We know that because we measure them as well with nanocytes, but they are not CD63 positive. Okay, so again, the importance of multiplexing to actually avoid negative results. Okay, so then we moved on, like, okay, if we produce exosomes from these cell lines, what is the representation of these panels and markers? So if we look, if we run the Quantigene assay, which is using the median fluorescence intensity, using EpCAM, cytokine 19, FN1 and HER2, SKBR, it's positive for all of them. But the exosomes, you can see cytokeratin 19 and HER2. Okay, so same trend, but at a smaller scale. Okay, but you still actually can detect them. Whereas HS570, which is in this case for this particular panel only positive for FN1, you also see the same trend in the exosomes which are derived from this originator cell. And when you compare 40 different genes in a multiplex fashion, and here we have identified and created panels which we call CRC AMP for amplifications and diagnosis, CRC invasion, which are important gene amplifications and the result involved in invasion, and CRC METs, which are in metastatic disease. So when we put them together, then we find those that are amplified. In this particular cell line, we have these which are amplified, and you can see that if you plot the MFIs from 250 nanograms of RNA, it has a very nice regression with MFIs resulting from exosomes. Now, of course, you find some outliers, but in general, for gene amplifications, you will actually have this nice correlation. So yes, we can actually measure the gene amplification in exosomes that originate from specific. Well, we didn't stop there, so we actually have set up a method, a workflow to actually isolate them now from patients. In addition, we also can detect the AMPs in 0.5 millimeters of FFPE cores, and this we showed the result before, primary versus metastatic tissues, in which we found these different gene amplifications in metastasis. But of interest, we managed to detect these particular 4 gene amplification in some of our patient-derived exosomes using only 0.5 mils of plasma. And this is an effort between our university and Omnigene, in which we are setting up this simpleplex assay to actually isolate these exosomes and read them from patient plasma in CRC. So what we are currently, what I've put together here is, we're starting from biology. We understood that there is a specific phosphatase, and we can actually look at specific markers. And those markers, we went into the interest of gene amplifications because it was involved in gene amplifications. And therefore we wanted to read cellular biology at a tumor level, but also in the tumour-derived vesicles and cells in blood. So we know that there are various clinical unmet needs, like equivocal diagnosis, heterogeneity of tumors, looking at the dynamics, progression of disease and resistance to therapy, okay, There are also problems to validate novel biomarkers, and therefore multiplexing and this high throughput methods can be used to actually do that from various sources. And of course, we're put together an enabling technology together with Thermo Fisher, Luminex as enablers, and we go after high accuracy, precision. Most importantly, and very important, is that we are getting a digitalized result, okay, in which we actually can also use it for patient surveillance, because we can read less than 15 cells, we can read exosomes now, we know as well. Okay, we can measure markers in low quality material and low input from FFPE. So this is all done and tested. So for these studies, it was only possible through the efforts of various parties, including the industrial partners, the enablers, Luminex for the xMAP technology for the multiplexing, Thermo Fisher for their Quantigene assays, Omnigene for putting together workflows for the exosome research, then the whole research group, including Christian Sherry, Janice Sherry, Michelle Cecchi, Istvan Mifsud, Jessica De Battista, the clinical consultants from the Pathology, James De Gaetano, from Surgery, Charles Cheney, and the navigators who took care of all the consenting, and of course the patients that consented to the study. And of course, all this cannot happen without funds, therefore thank you to the grant holders, Malta Council for Science and Technology, and the RIDT Fund and Alive Foundation. And with that, I thank you, and I open the panel for any questions. Thank you very much.